Okay, I want to first thank the organizers for the kind invitation. According to the program, I have 1 more minute left for my presentation, but I hope that you'll be able to stay a little bit longer. It seems like a big shift of gears from the previous presenter who painted a very, very broad canvas. Now, to make the link in natural products, we take up the challenge of using only a tiny part of Mendeleev's table. We work just with carbon, hydrogen, oxygen, and a little bit of nitrogen. But nonetheless, the number of combinations is, of course, astronomical, because we have molecules that contain, tens and tens of carbons and so on. So the number of natural products is probably almost unlimited, which is, of course, one particular challenge. We are not so much interested in the intrinsic properties of natural products, which may be interesting, like their UV absorbance, for instance, but we are interested in how natural products interact with biological systems, their bioactive properties. And so I've worked for a big part of my life on natural products and how they interact with certain biological systems to obtain effects of interest, like if they interact with humans suffering from a disease, whether we can improve the symptoms of the disease or even cure it, not just in humans, but also animals, veterinary applications. So the presentation I've chosen is just one small topic that we worked on, namely the effect of natural products on shrimp in aquaculture. So I'll start with a general introduction, which were the objectives at the start of the project. We had the preliminary screening of a number of plants. We used bioassay-guided purification to find the active compounds, and then we tested some of the plants in shrimp, and at the end I'll come to some general conclusions. So shrimp are an important source of proteins. You know that with the increasing number of people on this planet, feeding them and giving them a high quality diet is a challenge. And so aquaculture, and in particular shrimp aquaculture, is seeking to make a contribution to that. And it is of growing economic importance. Most of the shrimp that are grown in aquaculture are either white leg shrimp or giant tiger prawns. They represent approximately 90% of the shrimp production. As you can see, over the years, shrimp production has increased, and it continues to increase. There's been a small dip due to COVID. Most of it is located in Southeast Asia. You see China, India, Vietnam, Indonesia, Thailand. But it also is cultivated to some extent in South America, like Ecuador, and more recently also in the Middle East, a region not exactly known for having a lot of water. Now, there are also problems with this aquaculture because in contrast to the ocean, where most of the shrimp tend to stay relatively far apart from each other in aquaculture, they're all squeezed together in a small tank. And so infectious diseases can really create havoc in those environments. They can cause not just local problems, but some of these infections can cause real pandemics, where they spread worldwide and really leading to a complete collapse of the shrimp aquaculture industry. This has already happened. So there are two major types of diseases. One is bacterial ones, mostly caused by Vibrio species. The term is the disease called vibriosis, but there are different Vibrio species, Parahemolyticus, Harvey, Alginolyticus, Vulnificus, Anguiarum, and Splendidus, that can cause disease outbreaks. On the other hand, there are the viral diseases like the white spot virus that can cause severe problems. Now, the Vibrio disease is called the acute hepatopancreatic necrosis disease, usually abbreviated HPND to speed things up, or early mortality syndrome. Is caused by Vibrio species, which is a gram-negative bacteria that likes to live in conditions where there is salt water. It causes acute mortality amongst the cultivated shrimp approximately 35 days after stocking. And the signs here you can see a shrimp with disease and a normal shrimp. So the stomach is empty because it stops eating. The liver and the pancreas are basically destroyed, and the stomach in the normal shrimp is full. And also the rest of the gut is empty because, as I said, they stop eating. The most common viral disease is a white spot syndrome disease, and this is caused by a virus, a fairly large virus that has an envelope. This contains double-stranded DNA. It's very virulent and also highly lethal. So within a week or so, you see that 80 to 100% of the shrimp will die, and the shrimp show these typical white spots. They also stop eating and they swim more slowly. So the question we asked is, can we do something about it? Now, what most shrimp farmers do is they use antibiotics. Now, this, to begin with, makes no sense for viral diseases, but okay, they don't always know what is the problem. And even if it's a bacterial disease, the use of antibiotics poses serious problems. The more antibiotics you use, the more you select for resistant strains. And these resistant strains don't stay in the ponds where the shrimp live. They are also transferred via the humans that come in contact with them. And so this antibiotic resistance in the bacteria of the shrimp lead to also antibiotic resistance in human bacteria, which renders the treatment with antibiotics less effective. And in most hospitals in the world these days, every day patients are dying because we've run out of antibiotics to treat them. There are no more antibiotics that work in some of these patients. And the problem is increasing. That's why if you try to sell shrimp, they're tested for the presence of antibiotics. If there are residues of antibiotics found, at least when they're sold in Europe or the United States, they are just thrown out. They are not fit for human consumption. This is, of course, a big economic loss for the shrimp farmers. So they're looking for alternatives. And we have looked in this case, but also, as I said, in others for human and animal diseases. We've looked to plants as an alternative for the traditional antibiotics. Why plants? Because many plants produce secondary metabolites that have antimicrobial effects. They probably don't do that to please us. They probably do that for their own defense, because they also have to defend themselves against the bacteria and viruses. Now, since time immemorial, humans have made use of the presence of these chemicals long before synthetic chemists could make any organic molecule, to use these plants for their bioactive compounds. And so in several regions, world traditional medicines have developed so that in many of these plants, we know that they can be safely consumed by humans. At least there is no acute toxicity. The long-term effects are oftentimes less clear. And also shrimp farmers have at times used the plants to combat these infections that they encounter. But usually there is very little scientific evidence to support these practices, which does not mean that they're ineffective. It's just that there is no formal proof. The plants that they use are oftentimes locally available for practical reasons. But that means that they're accessible even to small farmers that don't have deep pockets, that the production of these plants can be made sustainable, and typically the natural products in these plants that are, have the antimicrobial effect are readily biodegradable. So they're not like these PFAS molecules that will probably stay in the environment for centuries, so they don't accumulate. And in most plants, you don't find a single antimicrobial compound. You typically find multiple antimicrobial compounds, and these combinations make it much more difficult for pathogens to develop resistance against a combination of antimicrobial components. Moreover, these components are typically different from the traditional antibiotics that we use, so there is less chance of cross resistance, and it's possible to use them while keeping the antibiotics we use for treating humans effective and usable, hopefully, far into the future. So when we started this project, our objectives were, in general, to help the shrimp production, reducing mortality, and find a herbal remedy against selected pathogens. Specifically, we would try to reach this objective by screening a number of plant extracts against these Vibrio pathogens, to isolate the bioactive compounds from these plant extracts by following the bioactivity during the fractionation. And we would also test these extracts, and if possible the compounds, for their effect against these pathogens of shrimp. Moreover, we planned to, once we had isolated the individual compounds, to also test combinations of the compounds and perform toxicity studies on them. Okay, so the first step, we need to select some plants. Which plants do you start with? Because you want to enrich, of course, your selection in plants that are more likely to contain effective compounds. We did this based on a literature study. We selected about 60 candidate plants from Bangladesh, and we prepared first small-scale extracts in different solvents, tested these for their effects against 6 Vibrio species. Then we tested various doses to see where was the concentration that had 50% of maximum effect, is a measure of the potency, and selected then the most promising plants for large-scale extractions. Here you see some of the results. So these are the 6 Vibrio species that we tested, and we plot the number of extracts that were active against these different species. So you see that some of these Vibrio species, like the Vulnificus and the Splendorus, are quite susceptible. Many extracts have an effect, whereas for some, like the Parahemolyticus, which is really the most important pathogen, far fewer extracts have an effect, so these are more resistant. You also see that the hexane extract in red, few of the plant extracted in hexane showed activity, and most of the extracts that are active are either extracted with ethanol or with acetone. Here is a so-called heat map of the results. So what you see here is all the plants that were tested, and in the other direction you see the extracts and the various Vibrio species that they were tested against. The color code is red means the extract was active, and blue means it was inactive. So you can see here again that most of the hexane extract showed very little activity, and most of the activity is in the ethanol and acetone extracts, which are particularly active against these 2 species that are susceptible, less against the more resistant species, but even there you see some activity. So this was the first step. Based on that, we saw some clusters of plants that had interesting activity, and from these clusters we selected a few representative plants, and I will discuss 3 of them. These plants were then used for what we call bioacid-guided purification. So we start with a large-scale extract. About 200 grams of dried plant powder is extracted in about 2 liters of solvent repeatedly. Then this extract is dried down, the solvent is evaporated, is loaded on a silica gel column, and then eluted with a step gradient of solvents of increasing polarity, yielding a number of fractions. These fractions are tested against the bacterium in question, and the active fractions are then separated by HPLC on a C18 column and eluted with the water acetonitrile mixture. Typically, this yields 60 fractions, which are again tested against the same pathogen, and then the active peaks are basically separated again on ultra-high resolution liquid chromatography coupled to tandem mass spectrometry to identify the compound. So here you see some results. So this is these are the fractions from the silica gel column. This is the activity, so there are some fractions that give 100% inhibition. There's a first group, a second group, and maybe some individual fractions here. This is the result for another plant, Tectona grandis, where you see there's a group of fractions with complete inhibition, another one and another one. So from these groups, we then go on to this is the result from the third plant, Syzygium cumini, where you see that most of the activity does not appear early in the column when we elute with relatively apolar solvents, but starts to appear rather later when we elute with more polar solvents. As I said, the next step is to take the active fractions and separate them further using HPLC. And here you see from the Piper Betley, for instance, fraction 33 is separated further. Just a few peaks appear, and the peaks with blue arrows are the peaks that show activity. So sometimes you see that all the major peaks show activity. In other cases, you see quite a few peaks, including some big ones, but the active ones are actually some of the smaller peaks. So these peaks are collected and identified by tandem mass spectrometry. And in the case of Piper Betley, we isolated 2 active compounds, cinnamic acid and 1-hydroxycarbonyl acetate, shown respectively here in their chemical structures. All of them show activity on the different Vibrio species that they were tested on. In some cases, this activity is better than traditional antibiotics like amoxicillin, which has no activity on these 3 Vibrio species. But as you can see, the concentrations needed to obtain activity are quite a bit higher than what you typically see for commercially available human antibiotics. So it means that these compounds are considerably less potent than the antibiotics that are used for humans. In the case of Tectona grandis, the second plant, we identified 3 active compounds shown here with their chemical structures. And you can see that, again, there's broad activity against all of the Vibrio species, and the numbers are already almost an order of magnitude lower. That means that these compounds are considerably more potent than the ones that we isolated from Piper Betley. And the third plant is Syzygium aromaticum. There we isolated 5 compounds that were active with, again, activities ranging between 10 and 200 microgram per ml. Now, this is the chemical identification of the active compounds. This is of interest, but the bioassay was activity against a bacterium in a test tube. So the question is, will these plant extracts also do something when you test them on the shrimp? And so we added these plant extracts to the regular shrimp feed at concentrations of 0.2%, 0.1%, 0.05%, and there was also regular shrimp feed with no extract added as a control. So we have 4 different treatment conditions. The shrimp were fed these 4 different formulations, of course in 4 different groups, during about 4 weeks. And after 2 weeks they were challenged with the Vibrio, so they were infected with Vibrio to see what would happen. Now we monitored a number of growth parameters like their weight gain, the growth rate, the feed conversion, which is very important, is the amount of weight they gain for a certain weight of feed. And of course the farmers like to use as little feed as possible for a certain weight gain. So the feed conversion is an important parameter economically for the farmer. We also monitored a number of immune parameters just to see what would be the mechanism by which the extracts would act. And so what we found is that for 2 of these plants, Piper beta and Piper emblematica, that they actually, compared to control, they increased the growth rate significantly and did so with the improved feed conversion. So you see that the feed conversion is better than the control. So this is very nice. The shrimp grow better even though they consume less food. And for 2 other plants that were tested also in shrimp, you can see the same, that compared to controls they grow faster and the feed conversion is better. So these are attractive properties from a commercial point of view. Now the shrimp were then challenged, that is exposed, either to the bacteria, the Vibrio, or the virus, the white spot. And what you see here is that in the control condition where the shrimp have not been treated or have just received a regular feed, the mortality goes up to 100%. So they all die. All the shrimp are dead at the end of the experiment after 14 days. In the case of the plant extracts that were tested, sometimes different dosages, you see that there is a certain protection, and so the mortality is less. And this is the negative control, so even in the shrimp that are not infected, there is some mortality, but we're not quite reaching that level, even with the best of treatments. This is the same kind of graph but for the viral challenge. So again, if you just give the shrimp regular feed and infect them with the virus, they will all die within a few days. They're all dead. Whereas when you treat with certain plant extracts, you can significantly reduce the mortality. Here we're approximately at 20% mortality. Still significant, but much less. So there is a benefit of treating these shrimp with these plant extracts. Now, maybe you didn't know this, but these plants were selected based on their activity against Vibrio, this bacterial pathogen. But some of these plants also seem to protect against a virus, which is a bit unexpected because the activity against Vibrio cannot explain why the shrimp are also protected against a viral infection. So it's probably not only a direct effect on the bacteria, which we have demonstrated and which is there, but it's probably also a general effect on the immune system, of the shrimp. Their immune system is probably stimulated, and that makes them more resistant, not just against bacterial infections, but also against viral infections. And we have some evidence to support that. So these are some of the parameters that give us a view of the immune system. So hemocytes are cells in the shrimp that are part of the immune system that defend the shrimp against invaders. And what you see is that indeed, when you treat these shrimp compared to control, the number of hemocytes increases significantly, both before and after the challenge. And the same happens with some of the other plants. So there is some evidence here that at least components of the immune system are stimulated. The hemolymph clotting time is another parameter that shows an activation of the immune system. The phenol oxidase is an enzyme that helps to defend the shrimp against pathogens. So again, you see that there's an increase in the activity of this enzyme after treatment with the plants compared to the control. And superoxide dismutase is another enzyme that's involved in generating free radicals, which then have an antimicrobial effect. So we can say that, in conclusion, that several of the plant extracts demonstrated significant activity against a range of Vibrio pathogens that we used bioassay-guided purification to isolate the compounds from these plants, that they display broad activity against Vibrio, but that they also have effects on the immune system and thereby increase in general the resistance capacity of these shrimp against various pathogens, not just bacterial but also viral ones. And this suggests that these plant extracts can be employed therapeutically as an immunostimulant for shrimp in aquaculture. So in closing, I want to acknowledge the people who did the work. So most of this work was done by a talented graduate student, Alokesh Kumar Ghosh, who is currently a professor in Bangladesh. And he was supervised by a postdoc in the lab, Dr. Sujogo Kumar Panda, who is currently a professor in India. Alokesh Kumar Ghosh received the Prime Minister Fellowship from the government of Bangladesh to carry out this funding. He was affiliated at that time with the Marine Biology Division of Khulna University in Bangladesh, where he's currently now a professor. The shrimp experiments were carried out by the Shrimp Research Station in Bagarhat, also in Bangladesh. And at KU Leuven, we have to acknowledge the support of the mass spec facility that helped us to identify the compound. So I thank you for your attention. I'll be happy to answer questions. So maybe, okay. You want this microphone or? Okay.
Thank you very much for this nice conference. You inspire us. We learn a lot of things. First of all, I have to present myself. I'm Dr. Ali Zintin Bongera from Food Technology Department in the Higher National School of Agronomy. And is our topics actually that we were working on it. I have actually two questions. The first one is how the acceptance of the shrimp farmers about this new approach. That means they don't use maybe cheap antibiotics in their farm, and then they use plant extract. And the other things is the availability and that you give continuous availability of this plant extract to farmers of the year that don't use the antibiotics and they use this plant extract. Second thing also is the stability of plant extract, if we call it about metabolomics. That means maybe being lunch in one season, it's not for seasons, but if we use another plant, we change metabolics about season, and then can this impact the continuous use of this plant extract in sheep farmers? Other things is the economic issue. That means how to convince the sheep farmers about money to use this approach. Thank you very much for this.
Okay. Okay, these are lots of questions, all interesting ones and relevant ones. I'll try to answer them. So the farmers are, well, let's put it like this. Several of the farmers are open to the idea of using alternatives to traditional antibiotics, but because antibiotics for them are also a significant cost, and the antibiotics are also not without risk, and so it depends a little bit if they are cultivating shrimp for the export market. They know that they have to be very careful about antibiotic residues, otherwise, as I said, their products will simply be rejected. There's also increasing regulation. So in Europe, for instance, but also in the United States, the use of antibiotics in animal husbandry, so for farm animals, not to treat them for certain diseases, but the continuous use of antibiotics in feed at low dose is being phased out, and for some antibiotics is already forbidden. So, even if the farmers want to stick to the antibiotics, in the future they may not be able to buy them anymore. They may be forbidden in the longer term. But as I said, it's also not a good idea to use antibiotics for that purpose, and I think they're becoming increasingly aware of that. It also causes environmental problems, and I think in view of the idea of creating a more sustainable agriculture, hopefully the farmers will accept these ideas and go away from the use of antibiotics. Now, if you want to make them go away from something towards something else, an alternative, you have to make that alternative attractive. And so one way to make it more attractive is to make it financially attractive. So it has to be at a lower cost, or there has to be some additional benefit. Now, I mentioned the improved feed conversion ratio, which means that they need less feed to get the same weight of shrimp, or for the same amount of feed, they get more shrimp weight. So if we calculate that benefit, because we know the price of the shrimp by weight, then for some of the plants that are commercially available, the cost of the plant can be taken care of by the improved feed conversion. So it would be an operation where the farmer does not have to pay more. He could stop using the antibiotic, and from the improved feed conversion, there would be room to purchase certain plant extracts. So it's economically viable. Now, these plant extracts, as I said, the ones that I mentioned, most of them are commercially available. That is, if you go on websites like Alibaba, you can buy them, in big drums of 50 kilograms. Of course, if all the shrimp farmers in the world start using that, the production of these plants will have to be increased significantly. Now, most of these plants can be cultivated, so it is possible to grow them, but of course they would need to be grown at much larger scale to satisfy the market demand, if there would be a significant switch to these plants for use in aquaculture. The continuous use would of course entail a cost, but as I mentioned, it probably would not create a big selective pressure against resistance. So because, as I've shown, in most cases you find multiple compounds, and the probability of resistance emerging simultaneously against multiple different compounds is actually quite small. This is what we now use in medicine. In combination therapy, we don't treat, for instance, HIV anymore with a single drug. We treat it with a cocktail of drugs. Many cancers are treated with drug cocktails. So it's something that, as I said, has been accepted as a strategy also in human clinical medicine. So I think the prospects for this type of treatment are reasonable. It would also fit into the idea that we have to switch from, and okay, I'm trying to be careful because there are people from the oil industry here, but we have to, in the future, switch from a fossil fuel-based economy to a sustainable economy, which means that many of the chemicals that we now use will have to be produced in another way than the chemical synthesis starting from building blocks that we get from the petrol, from oil. So these natural products fit into that shift, so they fit into this idea of a green chemistry. Of course, growing a plant just to extract a few components that may constitute maybe 1% of the total dry weight of the plant doesn't seem very effective, because then you have 99% of the plant that's left and may even pose a problem. Of course, the other way of viewing this is that plants will be put into a so-called biorefinery. This is a concept that, of course, is very well known in the oil industry. They put in crude, and out comes a whole range of products. We have developed that over many decades and optimized it for oil. It's not yet a reality for biological materials. We can't, powder a tree and put it in a biorefinery and out come a whole range of products. But the idea is, and the first pilots have already started, that we would be able to actually do that. To put a very complex mixture of a plant extract into one of these biorefineries and separate it into all the useful components like secondary metabolites, but also sugars that can be used for fermentation, fibers that can be used for textiles and for construction materials. So, I mean, plants can be pulled apart into useful fractions, and that, of course, will help the economic arguments, because if you only have one component of a whole plant that has to sustain your economical argument, it's much more difficult than if you can find a use for almost every component of the plant. Now then, the last component of your question is, okay, plants, of course, they depending on the season, the precipitation, the sunshine, they don't always have the same content of the active compounds that we're interested in. We can also use that to our advantage. I mean, via selection, for instance, we can increase the amount of the compound of interest. You may know that for cannabis, for instance, the selection by people who smoke this and who grow it has led to over 10-fold increase in the levels of THC, tetrahydrocannabinol, which is the active compound in marijuana. So it is possible to find cultivars and select cultivars that have a higher yield. Also, the environmental conditions can sometimes be adjusted. Of course, we don't control the sun, but we don't control the rain, but we can water the plants. We can protect them from the sun by shade. We can give them the right nutrients. So there are ways to improve the yields from the plants via genetic selection, via changing the conditions to improve the yield. Also post-harvest, the post-harvest treatment can also help a lot to preserve the components of interest and avoid their degradation. So I think in cases where it has been tried, there are some success stories. So I think we can use those as a guidance for the future. And if we're not going to produce these natural products in plants, we can now genetically engineer other organisms to produce the same compounds. So if the plants are not an economically viable way to produce them, then we can envisage producing them using the same biosynthetic pathway, which we could just transfer from the plants into bacteria, yeast, or other organisms that can be fermented on industrial scale. So I hope I've answered most of your questions. Okay, any other remarks or questions? I'll be available for the rest of the conference, so if you don't want to ask a question in public, come and see me during the breaks. Okay, you have, you want the microphones? Okay.
So my question is, how did you do the separation of the different compounds of extracts? You did the analysis by CMSMS, and how you reproduce this extraction, such you know, the most separation in CMSMS is the second part of the MS. So how did you reproduce this in the column, like you explained, to get the compounds separately and do the experimentation on the field? So this is my first question. And the second one is, how did you did you practice toxicology analysis or toxicology assessment of this experience, and is it on laboratory or on the field? And if it's in the field, how did you measure the concentration of these compounds on the field grade? Thank you.
Yeah, okay. Okay, so again, several questions. So I did not say much about the technical aspects, but as you can imagine, a plant extract is a very complex mixture. It contains, several hundreds to probably a few thousand of compounds, depending on the level. And as analytical methods improve and detection limits get lower and lower, we find more and more compounds in the same extract. Now, the way to isolate the active compound is to use chromatographic separation methods to separate this very complex mixture into fractions that are simpler. Typically the first step in this chromatographic separation process is a silica gel column. I didn't show you a picture of it, but silica gel columns can become quite big on the industrial scale. They can be several meters tall and have diameters of up to 1 meter and more. So they have very high capacity or scalable. You can load a lot of material on such a column, and we elute them with a step gradient. So we use solvents. We start with hexane, for instance. So in the first round, all the components in the extract that are dissolved easily in hexane will be eluted through the column. They will be separated a little bit on the silica gel column, but the resolving power of the silica gel column is not very high. And then we move to the next step. So we start to use mixtures of hexane and more polar solvents. So we start with, for instance, 90% hexane, 10% ethyl acetate, 80% hexane, 20% ethyl acetate, and so we keep increasing the ethyl acetate till we're at 100% ethyl acetate. Then we start with ethyl acetate methanol mixtures. So in each step of the gradient, we start eluting more and more polar compounds that are then ending up in those fractions. Then those fractions are in a second round separated by HPLC on the C18 column. This is based on hydrophobic interactions. So it's a separation method that's not quite orthogonal, but it's separating the compounds on a different principle. So compounds that would not get separated on the silica gel column may get separated by HPLC. And as you could see, in some cases the chromatograms showed multiple peaks, so which means that these fractions still contain several compounds. You could also see that the peak sizes were different, suggesting that the amounts of the different compounds are very different. And when we can separate the compounds well on the silica gel, we can collect the pure peaks, which may consist of a single compound. In that case, we know that we're dealing with a single compound and it is active because we have tested it on the Vibrio. Then how do we identify it? We put it in an LC-MS system. So when the compound goes through the column, it comes out and is injected into a mass spectrometer via electrospray ionization. And then the mass of this molecule is determined very accurately. It's determined up to with an accuracy of almost 1/1000th of a Dalton. So these molecules are several hundred Dalton big, and we measure their mass with an accuracy that is 1 part in a million, 1 ppm accuracy. That means that based on the mass, we can infer the atomic formula, also using isotope information. And when we know the atomic formula, we can search in databases which known compounds have the same atomic formula. But the mass spectrometer does more than just determine the accurate mass. It will also, in the second stage of the mass spectrometer, the molecule will be fragmented. So it will be broken apart, and the fragments will also be measured as to their mass. So we know how many fragments there are, and we know the mass of each of the fragments. And that then leads to a sort of a jigsaw puzzle that you have to put back together. So if you know the piece of the puzzle, sometimes you can put back the original molecule. So with that information, the exact mass, the fragments, and databases of the natural products that are known, we can oftentimes identify with quite good confidence which compounds were present. If that's not possible, we have to purify more and identify the compound via NMR. But NMR requires much more material, and a material that is completely pure. Whereas in the mass spectrometer, basically, if the material is not pure, first of all, there is a UHPLC stage. Then there is the mass spectrometer that can separate compounds of different masses, even if they co-elute from the column. So you know, that's the way we separate and ultimately identify. Then as to the stability, we have not done stability studies ourselves, but for some of these compounds that are known, there is some knowledge about their stability. The toxicity, we have started to do some toxicity studies, mostly on aquatic organisms like brine shrimp, because that is where they will be used. But these days in Europe, for instance, you cannot bring onto the market a compound without extensive toxicity studies. So ecotox studies, but also human toxicity, animal toxicity, as I said, whether they accumulate in the environment or not. So all of these studies have not been done, and they will need to be done in order to commercialize these products, at least if you want to commercialize them as treatments. If you just want to add them to the feed, you could just say it's a component of the feed. Many animal feeds already contain plant extracts. You cannot then make claims about the added benefits of the plant extract. You just have to say, this is a better feed. And why that is, okay, you don't make any claims about it. If you want to make medical claims, you have to do the studies and have to show the activity that you claim is there. And I guess there was one other question that I forgot. Okay, so the commercialization I mentioned, you know, what is needed for commercialization. We're not there yet, but I think we've shown hopefully enough so that a company will get interested to further commercialize this.